Hello guys and welcome back to Excelsior Academy. So in the previous lecture we started with the actin filaments, right? So let us just recap like what we have learned through this video. So this video which we saw in the earlier lecture. So let's watch once again and let's revise. Okay. So about the ARP2, 3, profilin, cofilin. So there is a G-actin, globular actin it is, ATP. Now, ATP is bound to G-actin. And this is how the hydrolysis takes place. So ADP actin has got low association affinity. Now they're trying to form a nucleation. So at least 3 molecules of ATP G-actin is needed, onto which further the active or the G-actin will bind. So this becomes your negative and the other one becomes your positive end. So it keeps on adding here and it keeps on getting depolymerized from here. So this results in the actin treadmilling. Now these are the actin accessory proteins, profilin and cofilin. So profilin is helping in bringing the G-actin with the ATP. So to the already existing nucleus, the profilin will start adding the new G-actin. Now comes the action of cofilin, which actually makes a cut in the actin filament so that new ends are created for both addition and deletion. Okay. Now let's talk about the association of actin filaments with the plasma membrane. So here we will discuss about certain proteins called spectrin. Guys, I hope you remember when we were discussing about the RBCs, erythrocyte membrane, there we talked about spectrin, that it makes a connection with the plasma membrane. If this is your plasma membrane, so it makes sure that the plasma membrane is held, and it also makes connections with the actin filaments. Okay, so now in detail, we'll see how this is happening. Actin filaments are highly concentrated at the periphery of the cell, where they form 3D network beneath the plasma membrane. So as I said, in order to maintain its 3D structure, okay, over here, actin filaments are present mainly. Now, this network of actin filaments associated with actin binding proteins is known as cell cortex. Determines the cell shape, involved in a variety of cell surface activities, including movement. So as in the previous lecture, I was explaining that it's always the actin filaments which helps in the movement. So, suppose the cell needs to come this side, like it has to roll. So here new actin filaments will be formed and here it will be broken. And that's how you have this depolymerization, the action of your cofilin, etc. Now, this is the structure of spectrin. So spectrin is your tetramer. Okay, now it has got alpha chain and the beta chain. What is ABD? ABD is your actin binding domain. Actin binding domain. Okay, now you can see that it's a tetramer and how it is formed? Alpha chain plus beta chain, and to the beta chain you have got the ABD. Okay, similarly on the other side, alpha chain beta chain, and ABD is present. Okay, so it's a tetramer. So majority protein that provides a structural basis for the cortical cytoskeleton in erythrocyte is actin binding protein called spectrin. Okay, so although spectrin itself binds phospholipids in the plasma membrane, the major link between the spectrin actin network and the plasma membrane is provided by the protein called ankyrin. So I hope you will be getting all those, images which we have studied, like how spectrin, ankyrin, and then the other one is band 3, which is also known as your anion exchanger, or it is also your bicarbonate chloride exchanger. So from this only we have studied like how the CO2 is being expelled out in the lungs. Okay, so that's your band 3. There is an additional link between the spectrin actin network and the plasma membrane, which is provided by the protein 4.1, or it is also known as band 4.1. So this will bind to the spectrin actin junctions as well as to the cytoplasmic domain of the glycophorin. So this is all we have studied in the previous lectures when we were talking about the integral proteins which is present in the erythrocyte membrane. Okay, now let's see in the figure like how the spectrin, ankyrin, band 3, band 4.1, all these are associated. Okay, so here it is. So you can see your band 3 is present, right? Now band 3 is there. You have got the glycophorin. Okay, this is your actin filaments. Okay, just now what we saw. So this is your actin filaments. Now see, actin is associated with the spectrin, right? And spectrin is associated with your band 3. All this with the help of the ankyrin. Okay, now actin along with the glycophorin is associated with the help of the protein 4.1. Did you get it, guys? समझ में आया? Yes, Akshaya?
Yes, ma'am.
Yeah, but you need to remember also, okay? तो आप इस किसी एक चीज को बेस बना लो, like actin is there. Now actin is associated with spectrin. Actin spectrum along with the band 3 is because of the ankyrin. Ankyrin spectrum and glycophorin is because of your protein 4.1. Okay, so you can see glycophorin hai, protein 4.1 hai, protein 4.1 is along with the spectrin and the actin. Okay, and then ankyrin is there. So please try to remember like which protein is associated with the what protein. Okay. Now coming to this DMD, that is Duchenne muscular dystrophy. Guys, this is really important with the exam point of view, and that's why I'm taking this right now in detail. Okay, now there is a protein which is related to spectrin known as the dystrophin. Okay, now it is of very particular interest because it is a product of the gene responsible for two types of muscular dystrophy known as Duchenne and the Becker. Okay, तो Duchenne तो lethally होता है, उससे कम lethal आपका Becker है. Now, these are X-linked inherited disease, which results in the progressive degeneration of skeletal muscle, and patients with the severe form have DMD, that's the Duchenne muscular dystrophy, and usually they die in the teens or early 20s. So DMD, you must have heard also that these kids, they don't have that skeletal muscle, they don't have this good skeletal muscle, right? Because of which it is difficult for them to stand. Right, it is difficult for them to walk also, and eventually at their early 20s they will be in the wheelchair, but at the early 20s they start developing all those heart, kidney, liver disease, and eventually they die because of the organ failure. Okay, so like spectrin, dystrophin also links actin filaments to the to the transmembrane proteins of the muscle cell plasma membrane. Okay, so जो अभी हमने देखा like spectrin is bound to the band 3 and all with the help of the ankyrin and actin, वो सब same way dystrophin का भी वही काम है. Okay, so these transmembrane proteins in turn link the cytoskeleton to the extracellular matrix, which plays important role in maintaining the cell stability during the muscle contraction. Okay, so what we are saying right now is that dystrophin. So dystrophin is making connections with the transmembrane proteins, Okay, ये आपका, let us say this is your skeletal muscle cell, उसका plasma membrane है. It has got proteins present over here. Transmembrane proteins are there. Now these transmembrane proteins are making sure that the actin filaments over here and the extracellular matrix are in connection. Okay, so this will help in maintaining the stability of the cell during the contraction. Otherwise what will happen, आप कॉन्ट्रैक्ट करोगे, सेल्स आपके स्क्वीज होंगे या इधर उधर चले जाएंगे. बट इफ यू हैव दिस मेंटेनेंस, व्हेन यू हैव दिस प्रॉपर मेंटेनेंस, इट विल हेल्प इन द प्रॉपर कॉन्ट्रैक्शन एंड रिलैक्सेशन, राइट? बट since this dystrophin is not there, so your muscle, or so your skeletal muscle is unable to contract. Did you get this point? This last wala, Krishita, is it clear?
Yes, ma'am.
Yeah, good. So these dystrophins are making sure that these transmembrane are making connections with the actin filaments present here and the extracellular matrix over here to maintain the cell stability for during the contraction. Since dystrophin is mutated over here or it is not present, so this connection is not being able to be made. Okay, and that's why with the DMD, the patients are not able to contract. There is no contraction, there is no activity, and hence they cannot walk. Okay, now since, as I said, this is X linked recessive inheritance, so if you have unaffected father and a carrier mother, okay, then the probability of having affected son is 1/4. So unaffected cell X and Y, unaffected daughter, like because X and X is normal. Carrier daughter, if she gets this chromosome, then with this she becomes the carrier daughter. Now if you have got the affected son, okay, so having this chromosome here and then Y chromosome here, so this person or this son is going to be affected, so this son is going to have the DMD. Okay, now can any one of you tell me, like, why from the affected son it is not transferred to the next generation son?
Because it is X-linked, the man passes the Y chromosome.
Yes, correct. Absolutely correct. Then what about having an affected daughter? Like suppose this affected son marries a carrier woman. Okay. Then what is the chance that the daughter who is born is going to be the affected?
Both need to be carrier in case of daughter.
Sorry, say it again.
Both need to be a carrier.
No, so I'm saying that, let's say that this affected son marries a carrier woman. Okay. So X आपका यहाँ पे defective है और दूसरा आपका normal है. So I'm saying that why we don't see any affected daughter?
Because it is recessive one, no ma'am. It's X-linked recessive trait. So
no, when you have both affected chromosome, let it be recessive, but the daughter is gonna have the effect. Did you get it?
That's what I was saying. If in case one of them are affected and other one is not, then daughter won't show. But if it is carrier in case of both father and mother, then daughter would definitely show. Is that the case?
No, Krishita. This is not how we say. See, it's good that I brought this then. Like it will be easy for us to do when we start genetics. Okay. So what I'm saying is, see, when you say carrier mother, okay, it means that she is not going to show the disease. Okay, she is going to be normal. This is the meaning of a carrier mother. Okay, now when I'm saying that this is an affected son, यानी कि क्या, इसको DMD है. Okay, now he is marrying a carrier woman. So where you have got the X affected, यहाँ पे mutation है, X normal. Okay, now the chances, इनके क्या क्या बच्चे हो सकते हैं? What are the chances? So they can have X affected. This is affected, right? This one and this. Ye or ye. So here we have, this is one daughter. Second daughter is going to have this and normal. This is going to be your carrier daughter child. Okay. And then again you can have X and Y. So again you're going to have a affected son and a normal son. If this X and this X. Sorry, that's Y and normal. Okay. So the chances is that you are going to have the affected son and affected daughter. What I'm saying is that why don't you see in nature, okay, any girl having DMD? Tanvi?
Oh, ma'am, you said one of the girl was affected. Then how?
हाँ, ऐसा जेनेटिक्स कहता है कि ऐसा हो सकता है, बट दिस इज लाइक मैथ्स, राइट? कि आपने कैलकुलेट किया है कि हाँ, देयर इज अ प्रोबेबिलिटी दैट अ डॉटर विल बी बोर्न अफेक्टेड.
Ma'am, because it is X-linked and women have one more X.
अरे हाँ कृषिता, but here I'm showing you, right? Both X are affected. That's why the daughter is having the DMD. What I'm saying is that why in nature DMD daughters are not existing. And I have given the answer in the previous slide. Is this part is clear? Where the affected guy marries a carrier woman.
Yes.
Okay. Huh?
Yeah, ma'am.
It's clear, right? So यानी कि इसके पास भी एक क्रोमोसोम है which is having the mutation and another chromosome which is normal. X and X. अब जब इनके gametes बनेंगे, so what are the gametes here? X affected, Y normal. X affected, X normal, right? Now I'm gonna have the gametes. One with this and one with this. तो क्या क्या मिलेंगे मुझे? X affected, X affected. ये एक हो गया. X affected, X normal, right? Now another one, Y. Y with the X affected, Y with the X normal. So X affected, Y normal, X normal, Y normal. Okay. Now I know this is a carrier daughter. This is normal son. This is affected son. This is affected daughter. Okay. Now all these are clear. So what is the probability? Everyone is having 1/4 of the probability. Correct. My question is, we do see affected son in nature. Now when I say in nature, इसका मतलब क्या? हमारी सोसाइटी में आजू बाजू कभी ना कभी तो हमें एक DMD वाला लड़का दिखेगा, ठीक है? But we never see affected daughter. Okay, I'll give you the hint. The DMD starts showing its symptoms at the very beginning. The moment the kid is around, 5-6 months, like when they start crawling itself. So by 1 year, the parents will understand that their child is having DMD.
Ma'am, has it got to do anything with X chromosome inactivation?
No, that is something very, 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 very far, Krishita.
Maybe the child won't survive.
Absolutely correct. That's what I was saying, that DMD kids are not able to grow or not able to survive beyond 20. Below that, sorry, before that itself they die. Okay. And that's why there is no chance that this marriage is going to happen. Okay. And hence there is no affected daughter, because you don't have affected son getting married to someone and, reproducing it. That's why we never see this. But if a normal person marries a carrier mother, then this is possible. Okay.
So
Now,
that is a Duchenne one,
hmm.
the one which dies early.
Yes, yes. Both chromosomes are affected. See,
So that is Duchenne one.
when you say something X-linked recessive, ठीक है? Recessive यहाँ पे मतलब क्या हुआ? It is present, but it is not lethal to the organism, and that's why the carrier mother is surviving as a normal life. ठीक है? अगर मैं ये बोलती कि ये X-linked dominant है, okay, it means the moment this zygote was formed, this X-linked dominant will have its lethal effects. यानी कि agar this woman was X-linked dominant, this particular what they are showing is dominant, then it means at the moment this was born, or she was born, would have died. Dominant means उसी time उसका lethal effect दिखेगा. ठीक है? X-linked recessive means mainly they are the carrier. Unless until they are present in homozygous condition, the lethality of the mutation won't be seen. X-linked dominant means even if it is present in one of the chromosome, it is lethal. Did you get it, everyone?
So, ma'am, one more question is, so Duchenne doesn't exist in male? Is it so?
It does exist in males only. And they die after 20 or before 20 years of age.
Okay.
It's just that DMD males cannot pass to their future offspring. This is only going to happen if the mother is a carrier. Okay. So there are many genetic disorders which is just because the mother is a carrier. Okay. Now it is clear. The slide is clear. And the concept of X-linked recessive and dominant.
Yes, ma'am.
Yeah, cool.
Yes, ma'am.
Now, let's see. So you have got this whole muscle. Let's talk about one bundle of the muscle fibers. And in that one muscle fiber membrane we are talking, right? So in this, you see, here there are the proteins. These are the transmembrane proteins. And to these transmembrane proteins, dystrophin is present. Okay. Now it is making connection. Dystrophin is making connections with the actin filaments, and this transmembrane proteins are making connections with the extracellular matrix proteins. Okay, so this is like more in detail. Okay, so here you have got the dystrophin. This is your dystrophin. It is making connections with your transmembrane proteins. Here you can see it is again made up of so many other proteins are there. Now these are the laminin. All these proteins are making connections with the, like for example, like the collagen, which is present in the extracellular matrix. Okay, so this is in just detail. You don't have to remember all the names of the domains. That's okay. What you have to understand is that dystrophin is making connections with the your actin, okay, and with the transmembrane protein. This transmembrane protein is making connections with the extracellular matrix. तो हमने क्या किया है? हमने actin filaments and extracellular matrix के जो components है उसको connect किया है with the help of the dystrophin and the transmembrane proteins. Okay, only this much you have to understand the role of the dystrophins. Now, what causes DMD? It is a defect in the X chromosome, which results in the mutation in the gene protein called dystrophin, and dystrophin is the largest human gene known till now. Okay, so it's a protein in the cell membrane of the muscles. So if the protein is absent, dystrophin is not there, then it is known as DMD. And if suppose the dystrophin is partially functional, then it is Baker's muscular dystrophy results. Okay. Now, because of this absence of the protein, it causes creatine kinase to leak from the cell and excess calcium to enter into the muscle cells. And because of this, later the cells die and are replaced by the fat and the connective tissue. So what is happening? You don't have muscle cells. Contractions kahan se hoga? Bas fat deposition hota rahega wahan pe. ठीक है. Now, can anyone tell me what is the role of creatine kinase? हम जब भी अपना blood sample देते हैं, they also check all this creatine kinase and all. Any idea like क्यों check करते हैं? इसका क्या role है? Have you heard about this?
It's a biochemical marker
For?
and for muscles cell. If the cell is not functional, that if the cell is not functioning properly, then creatine kinase tend to shoot up in the blood.
Yes, yes, yes. So if I say that in the blood report, the creatine kinase concentration is high. Okay, so it gives a signal. Okay, the diagnosis is what? It is giving a signal to the doctor that there is some muscle injury which has happened. Okay, it might be a muscle injury or it might be a certain disease which is related to the muscles. It will also tell you. See, if suppose what happens is that it's extremely high, okay, and you don't know why it is happening. So the first thing doctor checks is about the heart condition. Okay, so every time the heart muscles are always in function, whether you work out or not, your heart is always in function. So suppose there is something wrong, okay, ek choti si bhi injury rahegi, so the level of creatine kinase increases, and that gives a signal to the doctor, hey, there is some injury to the muscle. Which muscle it might be? So sabse pehle hum heart dekhte hain, kyunki you know, if it stops beating, everything is gone. But rest of the part of the body muscles, you can check later also कि क्या injury है या क्या disease हो सकता है. Okay, so please remember, whenever you go for your blood report, आप किसी का भी देखते हो, creatine kinase level is high, it means there is injury to the muscle, and especially we check for the heart muscle. ठीक है, good. Now, what are the effects? So, as a toddler itself, if he starts, walking, बहुत slow रहेगा, muscle degeneration रहेगा, because of which they won't be able to, start walking fast or running and all. So this is how the condition is. They do try to stand and get up, but it is really very difficult for them to get up. And see, instead of, we say that the calf muscles should be very toned up and all, it should look good. But what happens is, in the case of DMD is that the fat gets collected over here instead of having a muscles. Okay. Okay. So this is just the extra information, like what is the condition, then finally why, poor balance, they won't be able to walk properly, all the muscles is going to be very weak, and eventually when everything is weak, what happens is that the rest, many fractures will happen. Jahan jahan pe thoda bahut bhi bones rahega, every time they will fall and there will be a fracture because it's so weak over there. Now, how to diagnose? So, of course, high levels of creatine kinase in blood, you can go for the electromyography, genetic testing, muscle biopsy. Now, there is this case. So, I don't know. I mean, of course, it is not related to the topic, but this, I just remember one of the cases. So, you know what is IVF, right?
Yes.
In vitro fertilization, right? So there are people like, the couples who are not able to conceive by the natural methods, they go for the IVF. Okay, so I hope everyone knows like how the IVF works. So there was this scam which was happening, okay, in India. So what is that scam? See, I see right now the conditions are better. Like people are helping, people are understanding like we should go for the daughters also. Like daughters are also important, right? More than sons. But if you know, or if you have seen in your distant relatives also, or nearby in the neighbors or in the society where you live, still there are many people who go for the sons, right? So you know that by the natural way of conceiving, there is no guarantee that whether it is going to be a son or daughter, because it is going to be 50-50 chance. But in IVF, from the fertilized egg, okay, from the fertilized egg which happens in the petri dish. So from the fertilized egg, you can actually understand like which egg is going to be the son or which is going to be the daughter, right? Because sorry, you can understand from the chromosomes, like whether Y is there, it means it is son. If it is not there, then it is a daughter. Now, what was the scam was, they said that before implanting into the uterus of the mother, they check the fertilized egg for all the genetic disorders. Okay. Now, genetic disorder, when you go and checking for the genetic disorder, it's legal. To identify whether it's a daughter or son, it is illegal. Correct. But when you go for the legal way, checking the genetic disorder, and if you carry out the PCR and you know that there is no dystrophin. For example, what do you understand by this? It's going to be a son or daughter.
Son.
Correct. It is going to be a son, but having DMD. Right? Now, there are two ways, like how this has become a scam. So in the name of the genetic testing, they go and they check which fertilized egg is going to be a son or daughter. ठीक है, उसके नाम पे legal तरीके से आपने genetic testing की, and they tell to the parents, of course, when they say that definitely it's gonna be a boy, they charge more, and the parents who doesn't want like to know, let it be anything, so they are charged normally and all. Now just imagine that they have got DMD. And they say that if you want a son, but the son is going to have DMD, but they can help, by inserting a gene for dystrophin so that the child is not going to have the DMD. So the cost is increased. Of course, at the end, the couple is going to have a normal, healthy baby boy. But the process, in the name of the genetic testing, in the name of being legal, they have done this scam. IVF का क्या काम होना चाहिए? जो couples naturally conceive नहीं कर पाते हैं, उनको help करना, right? But this is how the scam started. Later it was found out, but the problem was that the institute which was carrying out IVF and the couple which wanted, so they were like, गलती तो यहाँ दोनों की है, right? So कौन किस पे क्या case करता है? So that's why it was dropped, and further actions were not taken. Anyway, so that was one way by which, in the name of the genetic testing, this scams and all happen. Okay, so I just remembered because of this genetic testing word. You can also go for the muscle biopsy, or you can go with the MRI, etc. Now, it can be diagnosed prenatally, that is amniocentesis. यानी कि जब बच्चा आपके womb में ही है, that time also you can check whether any genetic disorder is present or not. Okay. The treatment, there is no treatment. Physical therapy is there. They give steroids and all, but everything is waste. It just increases the survival days. Okay, but it is not gonna cure it. Then what happens is that eventually the kid dies. Right now, many places this gene therapy is going on, where you can actually replay the dystrophin gene with the new gene. Okay, or you can with the exon skipping, stem cells, all they are trying in order to increase the survival rate of DMD kids. Okay, let's come back to the actin. So that's all about DMD. Whatever is required with the point of your exam point of view, everything has been covered. Please do remember, like, what is the function of dystrophin, which all protein it is connecting, and whether how it is X-linked recessive. Okay, please do understand that and remember. So let's go back and talk about actin. So now let's talk about the cell attachments. Okay, so just now we discussed like how dystrophin is helping in making the connections between the actin filaments as well as the extracellular matrix. Okay, now what are these regions known as? Like what exactly all these proteins which is involved in these connections? So most cells, they have specialized regions of the plasma membrane which forms contacts with the adjacent cells of the extracellular matrix. So right now what we are saying is that, suppose your cells are present like this. Okay, so we are going to talk about the connections these which is made between the two cells and between the extracellular matrix and the cell membrane. Okay, so these are the two types of connections which we'll be discussing in brief only, because there is a separate unit for this talking about the cell attachments, like where we'll talk about the types of proteins involved in this: cadherin, E-cadherin, CAM, NAM, selectin, P-selectin, all these proteins will be present. Okay. So these regions can also serve as attachment sites for bundles of actin filaments that anchor the cytoskeleton to the areas of the cell contact. Okay, so these attachments of actin filaments are particularly evident in the fibroblast, which is maintained in the tissue culture. So whenever we study anything related to the extracellular matrix, yeah, connective tissue, or kuch bhi hua, we always go with the fibroblast. Okay, it is really very prominent, like by looking at the cells itself, like how they are making those extensions. Okay, so this is how under the fluorescence microscope it looks like, and you can see like the fibroblasts is making connections, so they can actually protrude out a lot. Can you see these protrusions? Now this blue color things are the nucleus. Do you guys have any idea like with what fluorescent dye these have been labeled? Any idea? किससे label किया होगा? Have you heard of something known as DAPI?
Yes, ma'am.
Yes, ma'am.
DAPI, we say DAPI. It is always for your nucleus staining, where DAPI stands for, anyone who knows the full form? No? So it is your 4-6 diamidino 2-phenyl indole. Okay, so it's an indole. So let me also write for you. It is 4-6 diamidino 2-phenyl indole. Okay, so that is your DAPI, DAPI. Okay, so this is for your nucleus. Everywhere, all over the world, nucleus is always stained with the help of your DAPI. Okay, because this has got affinity for your AT-rich regions. Okay, so it is going to label over there only. Now tell me whether the stain will only be taken up by the live cell or the dead cell. Yes. Is my question clear?
Yes, ma'am.
Okay. Yes, Tanvi, would you like to try answering whether the DAPI will stain live or dead cell?
Don't know, ma'am.
Okay. Anyone who has done or who has little experience in the animal cell culture?
I feel it should be dead
Okay, why do you think it should be dead?
because it is staining nucleus, and that's what I think. I do not have an idea about it, but I feel it should be dead. So.
Even
Okay.
I flip dead cell.
Okay, so your gut feeling is correct. Okay, so it is gonna stain the dead cell only. See, obviously, if the cells are alive, We have talked about so many transporters. If anything external, any compound which is external, which is not part of your metabolic pathways, why the cell will take those particular component, right? Even if it enters, it will try to expel out or pump out. Did you get the point? So live cells will try not to take the stain. Okay, since they are in the petri dishes, they are fixed. First of all, the cells are fixed. Okay, and during the fixing, you know, the cells are dead. So in this case, in the Tapi staining, what we do, the cells are there, it is washed, it is fixed, then it is incubated with the dye Dapi for at least 1-2 minutes. So Dapi solution is actually transparent. Okay, it doesn't look blue in color, it is transparent. So you've stain it, and then again you wash it to remove the excess of Dapi, and then you visualize under the fluorescent microscope. So what do you see? Blue color fluorescing very nicely. Okay, now let's talk about this green part. So this green part is actually labeling your actin filaments wherever the actin filaments are present. Correct? Now these are tagged by something called annexin 5 FITC. Fluorescein isothiocyanate. Okay. So please remember, like for annexin 5-HC, so you can change its affinity. Okay, so right now they haven't mentioned like which protocol they have used for. If you want to label actin filaments, you can tag with the annexin 5-HC. For nucleus, it is DAPI. For nucleus, it is always going to be DAPI because it stains your AT rich regions. Again and again, I'm telling, right? So that's why it is always going to be in the nucleus. Is that clear? So this is how under the, the background of a black, you get all these beautiful fluorescing, fluorescing cells are there. Okay. And this is how it looks under the normal microscope. Okay. So there's this video, just watch it. This is really very nice. Oh God, so much of volume. Sound आ रहा था क्या? No?
Yes, ma'am.
Okay. The cells are dividing. The cells are dividing if you concentrate. You can see that the cells are also moving. So they make the stretch protrusions with the help of the actin filaments. They move and they keep on dividing. Okay. So how much time it took, like from few cells to become the, around 90-95% complete. So this was taken by, taking the videos of one of the petri dishes. So this stage is known as confluence. 90% or 95% confluence. Okay. So we say that this is like over. अभी यहाँ पे जगह नहीं बची है. So it means what? There is no space. And we know that in the case of the animal cells, it is depends on the contacts. So जितना ज़्यादा एक दूसरे के आसपास आएंगे, उतना ज़्यादा कम division होगा then. There is no space, so they will stop dividing. There is no media, they will stop dividing, right? So that's why it is known as 90% or 95% confluent, sorry. Okay, so it will take around 72 to 96 hours. As you can see here, 92 tak dikha raha hai, 92 hours tak. So almost 3 to 4 days to reach this stage. Okay, so kaisa rehta hai? We take a petri dish. Okay, small wala rehta hai, jisko hum log P6 kehte hain. Okay, the company says it as P6. 6 is the diameter, I guess. So in this, we add around 3 ml of your animal cell media. So if you know animal cell media, RPMI hai, DMEM hai. So you must have seen that pink color solution, right? So those are the media for your animal cells. So we put it over there. Then we saw that at the beginning, there was only 1 or 2 cells were present, right? And when you see, and then we keep it under the incubator. Okay, we don't disturb them at least for 3 to 4 days. हम बस बीच में 3rd day में या 4th day में आके देखेंगे whether it is growing or not. But of course, after 48 hours, you should also check if any contamination is there or not. Let us say that suppose there was a bacterial contamination. What will happen? The bacteria will take all the media and the cells won't be able to grow. So even for that, we need to check at least once in a day to see if any bacterial contamination is there or not. Now, if there is a bacterial contamination, what happens? आपको पूरा का पूरा discard करना पड़ेगा because it will then spread to the other cell cultures also. So many people, like many lab people, they have the practice of adding antibiotics in the media itself. Very few people are there who doesn't who they don't add antibiotics in the media because antibiotics again something which is foreign object or foreign component for the animal cells, right? So it shouldn't affect its growth. That's why many scientists believe that we shouldn't add antibiotics in the media. But then many people are there who believe that it is not going to affect the growth of the cells. So they do add antibiotics. So it's again depending on the protocol followed in the lab. Okay, so this is how the fibroblast cells looks like. If suppose दूसरे कोई cell रहेंगे, so they will have little bit of circular shape. Okay, yeah. Now, so see, such cultured fibroblasts secrete extracellular matrix proteins and then they stick to the surface of the culture dish. Okay, now अभी हमें देखना है ये कौन से connections है I was talking about, right? Like they make a connections with the extracellular matrix. That's how उसको पकड़ के वो आगे बढ़ते हैं. It is just like the friction. So our body makes a frictional component with the surface onto which we walk. And then we push it behind and that's how we're able to move forward. In the same way, they make connections with the help of the proteins and then they move forward. So they attach to the extracellular matrix by making a binding of the transmembrane proteins called integrins. Okay, so they secrete out integrins which makes connections, and the attachment, the site of attachment are known as focal adhesions. That also serve as attachment sites for large bundles of actin filaments called stress fibers. Now these are important, guys. Okay, stress fibers, focal adhesion, integrins. अभी क्या हो रहा है? Focal adhesion क्या हो गया? It is the site at which this is happening. Okay, integrins kya hai? Produced by the cell. They are present over here, which makes connections with the extracellular matrix. Okay, and yahan pe aapke stress fibers hain. So these stress fibers, I don't know whether I've shown the figure or not. So they are like this, straight actin filaments like this. The bundles ऐसे रहेंगे. ये आपके stress fibers रहते हैं. So what happens is that when a cell is there, so these stress fibers will come like this, pushing the cell so that it starts moving forward. Okay, whenever I was talking about actin filaments helping polymerizing, so यहाँ पे क्या होगा? Stress fibers में इस side में आपके actin filaments आपके और बनते जाएंगे, ठीक है? And जो पीछे की तरफ है cell के at the back, So there the depolymerization of these stress fibers or the actin filaments will start. And that's how more and more, जैसे जैसे ये आगे बढ़ेगा वैसे वैसे stress fibers और आगे आएंगे. So all these are eventually helping in the movement of your cell. Did you get the point, guys? The importance of integrins, focal adhesion, and stress fibers. I'll show how stress fibers looks like. So stress fibers, they're nothing but the bundles of the actin filaments, which is cross-linked by alpha actinin. Please, guys, यहाँ पे ध्यान देना. So ये stress fibers हैं, they all are connected with each other with the help of alpha actinin and stabilized by tropomyosin, which will anchor the cell and exert tension against the substratum. Okay, we'll come to that. They're attached to the plasma membrane at focal adhesions with the help of the integrins. These complex associations are mediated by several other proteins called tallin and vinculin. For example, both tallin and alpha actinin bind to the cytoplasmic domains of the integrins. अभी हमने बहुत सारे proteins के नाम ले लिए. Let's go one by one. So stress fibers, okay, they are the bundles, and each इनको पकड़ने के लिए, see, when we have got the bundles of something, we wrap it with the help of the rope or any thread, right? So they wrap among themselves with the help of the alpha actinin and the tropomyosin. Okay, now जहां पे ये attachment होता है, ये cell हो गया, ये आपका extracellular matrix हो गया, that attachment site is known as the focal adhesions, and this attachment is done with the help of the integrins which is present, and this is done or carried out with the help of the tallin and vinculin. Okay, was I fast? Did you guys able to catch these words at least? I'll show the figure and then it will be more clear. Should I proceed? Are you able to follow, Akshaya?
Yes, ma'am.
Okay, let's see the figure. It will be more clear now. Okay, yeah. So look it over here. So we have got this extracellular matrix. Okay, now this is a plasma membrane. ये आपके cell का plasma membrane हो गया है. So just imagine your cell to be like this, ऐसा. And this is your extracellular matrix. Okay, now what I said, that at the focal adhesion there is a connection between the cell and the extracellular matrix. Correct? तो ये आपका हो गया focal adhesion जो हमने अभी देख रहे हैं फिगर में. Now integrins are there. So integrins क्या है? ये आपके cell membrane में present है. Okay, these are produced by the cell. Integrins are making connections with the extracellular matrix. Okay, did you get it? Integrins are making connections with the extracellular matrix. First point. Second, these integrins are in connections with the actin filaments. ये क्या है? Line wise इतने सारे, they are actin filaments. Since they are connected to each other with the help of the alpha actinin. Can you see alpha actinin? They are actually the stress fibers. Okay, so actin filaments connected to each other with the help of alpha actinin. So this is your stress fibers. They are connected to the integrins with the help of tallin and vinculin. Okay, now is it clear? This figure should be in your mind. Okay, itna clarity hona chahiye, because then only later when we'll talk about the cell movement, okay, we need all this proteins function to be remembered, guys. Okay, so Krishita, would you like to try explaining to us?
So there is this integrin which is attached to the extracellular matrix, and it is the one which is also connected to tallin and vinculin. And then there is this actin filaments, which actinin, alpha actinin, which kind of brings together those stress fibers.
No, alpha actinin is bound to the actin filaments. Together, these are known as stress fibers. Okay.
Okay.
Integrins are not connected to vinculin and tallin. Integrins are connected to the actin filaments via tallin and vinculin. There is no direct connections of integrins to the actin filaments. उसको connect करने के लिए हमारे पास दो protein है known as tallin and vinculin. ठीक है? So now you understand that अगर इनमें से एक भी protein में कहीं सा भी mutation रहा, what will happen? The connections or the focal adhesion, जो site of attachment है, it won't be there. The cell is not going to have its property, right? It won't be able to maintain its shape. When there is no shape, it won't be able to function properly, and hence that tissue is not going to work. When this tissue is not going to work, the organ is not going to work. Okay. So this is the fluorescent micro image or micrograph, the image of the stress fiber. This is how it looks. So you can see that there are the long stretches of the actin filaments. Okay. Now look at over here. So this is your cell, and you can see how nicely they form a bundle and they start, pushing the cells. So wherever the focal adhesion is there, the stress fibers are making connections and pushing the cells to move forward, right? So जैसे एक amoeba कैसे move करेगा? So even the movement of the amoeba is because of these stress fibers, the focal adhesions, etc., etc. Now please do understand like what are different types of the stress fibers. So here you can see we have 3 types. Dorsal stress fibers, which is present at the dorsal. It's a peripheric में जो present रहेगा, those are your dorsal stress fibers. Transverse axis, which is present on the top, ऐसे. Can you see this one? Yeah. And then the ventral stress fibers, जो आपके ऊपर से emerge हो रहे हैं. Okay. Just understand the location, and mainly it is the dorsal stress fibers which is helping in the movement. Okay. I think there are certain videos I have got. Yeah. Like how your stress fibers are moving. So what was happening? The cell was trying to come this side. Okay. Now this is again another image. Can you see the movement of the cell? ध्यान देना, ये जो cell है, right? So it's not like only from one. This particular region, it is moving towards that side. Okay, front की तरफ जा रहा है. And but what you will see is that the stress fibers moving ऐसे, जैसे पानी की तरह. Why? Because they are making sure that the movements are here the focal adhesions are there, right? So they will try to move in this direction, and what we see is that it is the continuous formation of the stress fibers, जो आपका polymerization and depolymerization हो रहा है, ठीक है? So keeping those thoughts in the mind, let's watch the video again. Did you get it? थोड़ा सा imagine हो रहा था, like all everything, like how the stress fibers are helping in the movement. Now this particular region looks very denser, right? This is nucleus. Yes. Any doubts? Further doubts? Tanvi, is it clear?
Yes.
Okay, let's move ahead then. 1 second. Yeah. So the actin cytoskeleton is similarly anchored to the regions of the cell-cell contact called adherent junctions. And these adherent junctions are mediated by another transmembrane proteins called cadherins. Okay. Cadherins forms a complex with the cytoplasmic proteins called catenins, which again will interact with the actin and the other actin binding proteins like vinculin, talin, which is anchoring the actin filaments to the plasma membrane. So अभी हमने क्या देखा? Already talin, vinculin, integrins, all these proteins are in connections. Okay. Now अगर मुझे दो सेल के बीच में करना है, then I'll talk about the cadherins. ठीक है, वहां पे हम adherence junctions की बात करेंगे, which is between two cell. अभी क्या था? Cell and the extracellular matrix के बीच में था connections, right? Focal adhesions we talked about. Now, if the connections are between cell to cell, then it is adherent junctions, and this is mediated by cadherins. वहां पे क्या था? Integrins. ठीक है? So there it was integrins, here it is cadherins, and here instead of having that those talin, vinculin, वैसे ही यहाँ पे catenins है. Did you get the difference? I'll just make a table, or you guys also can make a table. So it is between the cell and the extracellular matrix. Now this is between cell and cell. Okay. So there we talked about focal adhesions. And here it is about the adherent junctions. वहां पे integrins था, यहाँ पे cadherins है. वहां पे और other proteins were there like talin and vinculin. यहाँ पे catenins है. ठीक है? I guess I have the figure for this also. Yeah. This is between two cells. Okay. So imagine you are having a, ये एक particular cell हो गया ऐसे, और ये दूसरा cell हो गया. Okay, I have the figure here. Sorry. So there are two cells. Okay, हमने ये portion को zoom out किया है. So what do we see here? We have got those cadherin instead of integrins. Okay, जैसे वहां पे था आपके focal adhesions में, plasma membrane में cadherin. So this cadherin and इसका protein cadherin is making connections. Cadherins are attached to the catenin with the help of protein, glycoprotein 120. Okay, and they are making connections with the alpha beta catenin, vinculin together to the actin filaments. And actin filaments के बीच में कौन आता है? Alpha actinin as previously. ठीक है? Did you get the points here? So वहां पे था हमारे पास talin and vinculin between the actin filament and the integrins. Here we have P120, alpha beta catenin and vinculin, which is making connections with the actin filaments. Okay. I know it's a lot to remember, but it is also important to remember. Shall I proceed? Akshaya, is it clear? I hope at least the concepts are clear. Later you can by heart, you can remember. With practice, you can. Okay, shall I move? Shall I move ahead, Akshaya?
Yes.
Okay. Now coming to the microvilli. So they are the surface of most of the cells have variety of protrusions, and this kind of microvilli we see in the case of epithelial cells, for example, right? So they increase the surface area, and these cell surface extensions are helping in the cell movement, phagocytosis, or for the absorption of the nutrients. So in the case of the epithelial cells, we know that these microvilli are helping in the absorption of the nutrients, right? So on the epithelial cells, we have got these villi, right? And on the villi, we have got the microvilli. So they're like small actin filaments के protrusions रहते हैं, which is helping in the absorption. So here it is the here it is the electromicrograph of the microvilli, and they have taken from the intestinal cells. Okay, so all these line you can see here, they are nothing but your actin filaments, and नीचे जाके network बन जाता है. ऊपर क्या है? You can see that they are the straight lines, the bundles are present, right? Here the bundles are present, but at the bottom you can see it is the network. Okay, now let's see the one particular microvilli. So we have got the plus end. So plus end मतलब क्या? जहाँ पे you can add more of your actin molecules, monomers, right? Now these actin filaments are in connections with the plasma membrane. Can you see? What happened? Okay. Are you able to see the screen?
Yes, ma'am.
Okay. In between it was gone. Okay. So see the connections between the actin filaments and the plasma membrane. It is because of the calmodulin and myosin 1. What is calmodulin? It is your, calmodulin, it is your calcium binding protein. Okay, please remember. And myosin 1. What is myosin? Myosin is your actin filament ka motor protein. So when a microvilli is moving, okay, it is moving because of the myosin. So calmodulin and myosin is making connections between the actin filaments and the plasma membrane. Now, to keep these actin filaments in connections or held together, this is done with the help of the fimbrine and the villen proteins. And down you have got the network, this portion. It is known as the terminal web of actin filaments. Okay. Now, it is a movement of the myosin, the motor protein, which results in the movement of your microvilli like from here to here. ऐसे flip-flop होता रहता है. So that movement is because of the myosin 1. And this movement, for this movement, you need calcium. And that's why you have got the calcium binding proteins over there, which results in the movement. Okay. Did you get this? Then only I'll move ahead. So you have got the plus end. The actin filaments are held together with the help of the villen and the fimbrine. And these are in connections with the plasma membrane with the help of the calmodulin and myosin 1. Myosin 1 is responsible for the movement. Okay, it is your motor protein which moves or which is specific for the actin filaments. ऐसे ही microtubules के motor proteins हैं known as kinesin and dynein, which we'll talk about it later. And at the bottom, okay, here you have got the terminal web of your actin filaments. Clear? Okay. Okay, so now this is how on the top these microvilli looks like. Okay, so you can see like so many singles are there, and each one has got so many actin filaments ka bundle. Okay, now so microvilli, as we know, it increases the exposed surface area. So more surface area, more area available for the absorption. Now, in addition to the role in the absorption, specialized forms of the microvilli which is present in your ear. Okay, it is known as the stereocilia of the auditory hairs is helped in the detection of sound vibrations. So if you remember from our previous lectures, there were these stereocilia of auditory hair cells. They have got the receptors known as the mechanoreceptors. So whenever the air or the sound waves, okay, not the air, sorry, whenever the sound waves reaches your ear, okay, it falls or it makes this stereocilia to move. That movement makes your mechanoreceptors active and results in the giving the information or carrying out the, the calcium channel gets open up, which results in the passing the signal. And that's how your brain understands like, okay, ye sound aaya hai. This is what I am talking. This is what you are listening. Okay? So even there, it is the actin filaments which is helping. So these filaments in the bundle, they are cross-linked by the actin bundling proteins called fimbrine and villen, which eventually links the actin filaments in the closely spaced parallel arrays. Along their length, the actin bundles of the microvilli are attached to the plasma membrane by calcium binding protein called calmodulin in association with the myosin 1, which is involved in movement of the plasma membrane along the actin bundle of the microvillus. Now at the base, you have got the terminal web, where you have this actin filaments bound with the spectrin-rich cortex is there, okay, which is helping in stabilizing the microvilli. Same thing which just now we have discussed. Now here again. So this figure will tell you like, how two microvilli are present and how with the help of the proteins, it is helping in the movement. Okay, so can you see the protocadherin myosin over here? So what we are showing right now, what I want to say is that these myosin are in connections with the cadherin proteins. So cadherin हमने पढ़ा था अभी दो cell के बीच में रहता है, right? So they help the myosin protein to move around it. So what is happening? So here this is there. यहाँ पे जो proteins attached रहते हैं, like sorry, हाँ, here the proteins are there, right? So when they move, the microvilli move. Are you getting it? कैसे move होगा? I don't have the video here. So on the actin filaments, myosin is there, okay? This myosin. So when they move, if suppose myosin move कर रहा है, ऐसे let us say it is moving. What is happening? It is pushing your actin filaments downwards, right? If this is moving like this, it is pushing your actin filaments in this direction. So this is helping in general. Overall, the myosin movement will help in the movement of the actin filaments. अब इतना पूरा bundle of actin filaments जब move होगा, so क्या होगा? The microvilli will move. So up and down, you know, to and fro motions will come. Okay. थोड़ा सा idea मिला, like how all this are happening, how the microvilli are moving. Yeah. Okay. So that's all, guys. I'll be stopping here now.